Professor ERIK SEIFFERT, thanks so much for coming on to EvolutionSoup all the way from your office in California, USA. You are an evolutionary biologist and paleontologist at the University of Southern California, known for your research on mammalian evolution, particularly the early history of primates. Your research looks at how early mammals in Africa and Arabia evolved, adapted, and spread over 23 million years ago. Just recently, March 2026, you and your team announced the discovery of fossils in Egypt belonging to an ancient primate that you've named Masripithecus moghraensis, which may be the closest ancestor of all apes, including humans. Welcome to the show, Eric. Discoveries like this take a very long time to get to the point where it's published, so it must be gratifying to finally be able to present your findings to the world.
Yeah, it is. Thank you, Mark, and thank you for the invitation. Really appreciate it. The first really important discoveries were made on March 30th, 2024. So we actually managed to get the results published within 2 years. I actually was not in the field when the discovery was made, but my colleague Hesham Sallam at Mansoura University has very kindly kept me in the loop on his many discoveries that he makes in the field. So I often, due to the time difference between Egypt and Los Angeles, he's often in the field making really important discoveries, and I will wake up to a number of pictures that he has sent me of new discoveries in the field. Sometimes he's just, showing me a new fossil that is really cool. Other times he'll want my opinion on what has been found. And yeah, so this morning I woke up and opened up WhatsApp, and the first picture I saw was of these ape teeth, and very kind of peculiar ape teeth. But I knew right off the bat, and they already knew that they had found an ape. They didn't need me to tell them that. But it was immediately clear that this was going to be the first fossil ape that has ever been found in North Africa. So no matter what it looked like or who it was related to, just the fact that suddenly we were able to fill that really significant sampling gap was really a big deal. So fortunately, these days there are really good imaging options at Mansoura University, where Hesham leads a group of vertebrate paleontologists. So he was able to provide laser scans of the fossil, I think, within maybe 48 hours. And so I could immediately start to manipulate them and make comparisons. And it just so happened that I already had a matrix for phylogenetic analysis for determining relationships that I had already put together for a different paper. So it wasn't too difficult to just plug the new fossil into that matrix to run the initial analyses. So it came out in a very interesting position, and we can talk more about that. But we submitted it to Science. We decided, why not? We'll try for a major journal. We had a really positive first round of reviews, 3 reviews that all of the reviewers were very positive and had good suggestions. And we were able to meet all of their requests. And so it was published fairly quickly for a fossil discovery like this. And so it's great to have it out in the world, and it's great to have this option to talk to you, because I've noticed as time goes on, there's more and more of a gap between what scientists say in the papers, which are typically very measured, and then what you see in the popular media with headlines that are very sensational. And there's this huge gap, and paleontologists don't really get to talk about what we're really saying about our discoveries. And so this is a very important service that you're providing. Thank you very much.
Thank you for saying that. Before we learn more about moghraensis and its world, let's just hear a little bit about your background. Erik, can you take us down the road that led you to the research that you're doing now?
Sure. So when I was a kid, and through what we call middle school and high school here, I knew I wanted to get into some line of work that would allow me to travel and to discover new things and to experience other cultures and meet people in different parts of the world. But I wasn't really sure exactly what that would be. And going into college, I didn't know, where I would end up and what I would end up doing. But I think I was really fortunate that in the first year of college, I can't remember if it was my first or second semester, but I took a course on an introduction to biological anthropology course. And right off the bat, I realized that this is just amazing, and this is something that I could really get into. Then we started to go into human evolution and the trajectory of human evolution, and starting off with early primates, and then going into the origin of monkey-like primates. And the professor put up an image of the Fayum Depression in Egypt, which is where I've ended up doing most of my work in my career. And it's a picture that's not unlike the one that is behind me here. That is the Fayum Depression in Egypt. And in that moment, everything clicked. Like I just recognized that this is exactly what I want to do. And from that day forward, I was really obsessed. And so, as an undergraduate at Berkeley, I ended up taking courses in primate evolution from Isaiah Nengo, who sadly passed away a few years ago, but he was a Kenyan paleontologist who was making really important discoveries in the Miocene of Kenya. And that really sparked an interest in the evolution of apes, because that was what he was really focused on, is the evolution of apes, primarily in the early Miocene, but into the middle Miocene in Kenya. And I was taking courses in human evolution from Tim White, who of course is very famous in the world of paleoanthropology. And so from there, when I was applying to get my PhD to go into grad school, I really thought that what I would be doing is figuring out the relationships among all of the living and extinct apes. And so my first step, I ended up going to University of Texas in Austin, and started working on that problem. And frankly, got to be a little bit frustrated, because in ape evolution, there has been a tremendous amount of convergent evolution that makes it really difficult to figure out who is related to who. And also there's just a tremendous amount of variation within species. So it's really it is tricky to tease apart these relationships. Though more recently, people have made, I think, significant progress in that area. So it just so happened that this was also around the time when some really important discoveries were being made in the world of molecular phylogenetics, so DNA, genomic based phylogenetics, determining relationships among the living mammals. And yeah, I was about 1 year into my PhD work, and a paper came out on the molecular evidence that there was a clade, or a group of mammals that are more closely related to each other than to any other living mammals, that would come to be known as Afrotheria, which included a number of mammals like elephants, hyraxes, sea cows, aardvarks, mammals called sengis, used to be called elephant shrews, and tenrecs and golden moles. And these mammals, as I think most people can imagine, they really do not look like each other. And the title of the paper was "Endemic African Mammals Shake the Phylogenetic Tree," and it's because it was really a mind-blowing discovery. A lot of people were really skeptical about whether or not it's actually real, and so I decided that I had to do this instead, and so it took me down a different path. I decided to go to Duke University to finish my PhD, because Duke has a fantastic collection of fossils from the Fayum, lots of early members of this group called Afrotheria. And so I wrapped up that work at Duke, and that really, when I was at Duke, I also started to work with Elwyn Simons in the Fayum. And so I was going to the Fayum in Egypt every year. The first year that I was in Egypt, we discovered a new locality that would come to be known as BQ2, which is the oldest primate bearing locality in the Fayum succession, which goes from about 37 million years in age up to maybe 29 million years in age, so a very broad time window. And so that's kind of what got my career started. And then my first job was at Oxford in England, and I was a lecturer in the Earth Sciences department there, and I was also a curator in the Oxford Museum of Natural History. Was called the Geological Collections, but that's paleontology. So I spent 3 years there. It was a really good experience, but I then had the opportunity to move on to Stony Brook in New York to join, or to be a part of what was then a new initiative that was called the Turkana Basin Institute. So a research institute that was based in the Turkana region of northern Kenya. And Stony Brook was also very attractive for me at the time, because it was a vertebrate paleontology and paleoanthropology powerhouse. So many of the people who, I really looked up to, like Bill Jungers and John Flegel and Dave Krause and many others. And I had good friends and colleagues there, like James Rossi. And so it was just a very intellectually vibrant atmosphere at Stony Brook. And when I was at Stony Brook, I was teaching anatomy to students in the health sciences. And in 2008, I decided to make a move to Los Angeles here at USC, again in a department of anatomical sciences, teaching anatomy to medical students and continuing my paleontological research. And the goal here was to build up a program that would be similar to what has long been the case at Stony Brook. And we've built up a really good group here as well. And so yeah, that's kind of the trajectory that I've been on up until this point.
Your work focuses on primate evolution going right back to the Paleogene, a time period between 23 and 66 million years ago, which saw an incredible diversification of mammals as well as birds and plants. So, can you outline for us the research that you do and explain to us why, with this new fossil find, your work has come full circle?
Yeah, so as I mentioned, most of my work has been in the Fayum Depression of Egypt, and that's really the most important place in all of Africa for understanding what happened before the Miocene. So the apes and the monkeys, old world monkeys, apes and humans probably split from each other around 30 million years ago, but we don't really have a lot of evidence for what happened between maybe 30 million and about 20-ish million years ago in Africa. There's a fairly significant gap there. But there is this area in Egypt that is really spectacular. So as I mentioned before, it the deposits range in age from about 37 million, the oldest deposits, up to about 29 million years in age. So, and outside of the Fayum, there are some sites, but they just aren't very productive. Um, and so, like going even farther back in time, deep into the Eocene and into the Paleocene, stretching back now probably about 62, 63 million years back in time, we have snippets of information about what was happening there, including some primates, but we just don't know nearly as much as what we know from the Fayum. Typically, discoveries at other sites are isolated teeth of primates and the occasional mandible, little jaw fragment, or a maxilla, upper teeth. But in the Fayum, it's there are complete crania that are known of various early anthropoids. So anthropoids are the this is the primate group that includes the apes and humans, the old world monkeys, like think about like baboons and vervet monkeys and macaques. And then more distantly related are the platyrine monkeys of South America, sometimes called the New World monkeys. And those are like squirrel monkeys, spider monkeys, howler monkeys. So species like that have always been restricted to the neotropics. So their last common ancestor, and that would be the common ancestor of what we would call the crown anthropoids. So when I talk about a crown group, that means all of the living species and their last common ancestor. So anything that branched off before that last common ancestor would be what we'd call a stem member of that group. So we have crown anthropoids in Egypt. That means that they are within this clade and they're more closely related to one of the living clades, like the old world monkeys and apes, or to the platyrines, for instance. We actually don't have any relatives of platyrines in the Fayum yet, but they are conceivably might be found. And so that clade, the crown anthropoid clade, probably based on genomic molecular evidence, probably originated between 40 and 45 million years ago. And so we are in the Fayum, we're catching a later phase, probably after the initial diversification of crown anthropoids. And so we have the earliest stem members of the group that are called the catarrhines. So the catarrhines This is the clade within anthropoids that includes apes and humans and the old world monkeys. Okay, so their crown group, as I mentioned earlier, that they probably diverged from each other around 30 million years ago. And so we have a long stem lineage that extends all the way back to 40 or 45 million years ago. And we document those early phases along the stem lineage in the Fayum area. That would include genera such as Catopithecus and Oligopithecus, which are really primitive stem catarrhines that show the very initial changes along that stem lineage. For instance, losing one of the premolar teeth above and below, both the upper and lower jaw. And then we move, as we go through the succession, from 37 to 34 up to 29 million years ago. In the youngest levels, we pick up some species that look very similar to some of the early apes from East Africa. So these are species that are called Egyptopithecus and Propleopithecus, and they have molar teeth that are so ape-like that for many decades people actually thought that they were the earliest fossil apes. As it turns out, people came to realize that kind of tooth shape that is seen in these early stem catarrhines is probably the primitive condition within catarrhines, and we see a different kind of arrangement in the old world monkeys that is called bilophodonty, which means that you have two tall crests crossing the lower molars, again, above and below in the old world monkeys. And what we see with Egyptopithecus, Propleopithecus, and later apes, we have on the lower molars basically just 5 primary cusps and no tall crests that are connecting those cusps. So, but the Fayum deposits are really important for showing what was happening in Africa before we lead into this time period when the first stem apes were appearing, when they were first branching off of that stem lineage. This I always had a tendency as a graduate student and just in my career, I tended to want to, we'd find a fossil somewhere and learn about a fossil, and I always wanted to know what came before that species. And so I was always looking for to go back and back and back in time. And I actually went so far back that I started looking for fossils in the Cretaceous, before the Cenozoic. I didn't have any luck in the Cretaceous, so I'm now coming back, closer to the current day in the Paleogene and into the Miocene. But in that, as far as other primates that existed, it's important to point out that it wasn't just anthropoids that were present in the Paleogene of Africa. It was also, there was a group called Adapiforms, which are now completely extinct. And this is a group that was in the Eocene, was found in North America, probably right around where I live now in Los Angeles. There were Adapiforms. They were in Europe and they were in Asia. And there was one group that made it into Africa, probably in the Middle Eocene, that actually evolved, one of the lineages evolved features that was very highly convergent on early catarrhines. So this completely unrelated Adapiform clade had also lost their upper, one of their upper and lower premolars and developed what we call the third premolar that was very sectorial, so would have occluded with the upper canine. It was very obliquely implanted in the jaw in a way that is otherwise really only seen in catarrhine anthropoids. So a really fascinating example of convergence. There's a primate from this BQ2 locality called Nosmips that nobody has any idea where it came from. It's bizarre, and it's very rare to not be able to place a fossil primate in the tree. So we're still trying to figure out exactly what that is. But also we have fossils of the oldest fossil members of the group that are known as the strepsirrhines, that include the lemurs, lorises, and bush babies today. So all of these species very uniquely have what's called a tooth comb, an arrangement of the lower incisors that is used for grooming, but also in some species for gouging into trees to get, for instance, like gums and saps out of trees. And so we've been able to fill that gap as well, because prior to 2003 there was no record of this group in Africa, and it was always a huge mystery where they came from and how lemurs got to Madagascar, because if we weren't finding their ancestors in Africa, so how did they get there? With these new, well, not new anymore, but the discoveries that have been made over the last 2.5 decades, we were able to fill in that gap, and has really clarified what was happening with that group. And just generally, Africa, going back into the Paleogene and back beyond the Fayum deposits, it's a huge mystery for the most part. We can predict what kinds of mammals were there, but there's still a massive amount of work that needs to be done. Huge numbers of discoveries that will be made, I hope, in the coming decades. So yeah, so it's a long way of saying that I started off with thinking that I was going to study Miocene apes, went way back in time to figure out what was going on before that, and now I'm unexpectedly coming back into the Miocene to study that group again, the apes.
The new species was found in the Wadi Moghra fossil site in Egypt in spring 2024. Erik, can you tell us the story of the Masripithecus moghraensis discovery and why this species changes what we know about the origin of apes?
Yeah. So we're in this new phase of paleontological work in Egypt, which is really fantastic. Where Hesham Sallam at Mansoura University has this big vertebrate paleontology center, very very modern and definitely much better equipped than anything that I have here in the U.S. It's really an incredible situation that he has developed there. And so he has a number of students and volunteers in his lab, and they are going out to do field work quite regularly at different places all over Egypt. So in Fayum, in the area that I've talked about, they have projects in the eastern desert and the Paleocene-Eocene boundary, with particularly fossil fish that are being discovered, that it's a completely unique fossil fish fauna, and that is being that's the dissertation of Sana El Sayed, who's at University of Michigan working on her her dissertation. And there's work in the Cretaceous on dinosaurs and on crocodiles, and that work is being done by Hesham Sallam, who's at University of Ohio, and another PhD student who's at Oklahoma State, Abdullah Gohar, who's working on whales. So they're working all of these sites quite regularly, and just regularly making really incredible discoveries. And so there's also this site called Wadi Moghra, which is in northern Egypt in what's known as the Qattara Depression. And Miocene deposits have been known in that area for a very long time, going back into the early 1900s was when the first descriptive work came out, based on fossils that had been found in that area. And so now one of Hisham's students named Sharuk Al-Ashkar is working with Hesham and focusing more and more on the work at Wadi Moghra. And the nice thing about Moghra is that it's actually quite close to Mansoura. It's not a far drive. And new roads are being put in that make it easy to access a lot of these sites. It's also a little bit concerning because it means that people can very easily get to these sites and hopefully not make illegal collections. But so Hesham and Sharuk and several of the other people in his lab have been making regular visits to Moghra. And Moghra is it's interesting because it's about the same age. It's about 17 to 18 million years in age. So it's about the same age as some of the most important sites in Kenya. For instance, on Rusinga Island in Kenya, where going back almost many decades, there have been many important discoveries in Rusinga and multiple localities around Rusinga. But the primate faunas are quite different. So until this discovery of Masripithecus was made, the only primates that had ever been found at Wadi Moghra were Old World monkeys, and specifically a species known as Prohylobates tandii. So this was a stem cercopithecus, would have branched off before the origin of the crown group. So the crown group of cercopithecus includes the species I mentioned before, like baboons and macaques and vervet monkeys, but also colobus monkeys, leaf-eating monkeys. Their common ancestor probably split maybe around 15 million years ago, maybe a little bit earlier. And so Prohylobates that is in Wadi Moghra branched off significantly earlier than that, and represents a more primitive phase in the evolution of that group. So yeah, until this discovery was made, it was just Prohylobates and another unnamed species of cercopithecus. Whereas in Rusinga, everything appears to be either a stem ape or maybe a stem catarrhines, but there are Old World monkeys just generally in the early Miocene of East Africa are really very rare. So it's clear that by the early Miocene, there's already a significant biogeographic difference there between Northern Africa and Eastern Africa. I just had, kind of assumed that maybe more work in Moghra would just lead to more discoveries of monkeys. And so it was in 2023, actually, the first fossil of Masripithecus was found in, I believe it was 2023. and it was a jaw, but it was a jaw that was, all of the crowns of the teeth had been completely eroded away, basically wind blasted away, so you couldn't tell any of the details of the teeth. And Hesham had sent me a picture of that specimen to ask my opinion, and I didn't have a good sense for exactly how big it is, but it looked like it might just be another fossil that could be attributed to Prohylobates. It turned out after the Masripithecus fossils were found that is actually a jaw of Masripithecus, and we just hadn't really put that together yet. But it was an important piece of the puzzle, and it was very important for putting together a composite mandible that really helps us to understand what the jaw of Masripithecus looked like. So these fossils were found by Brook. Again, it was March 30th, 2024, and that got everything going, and we all started to work together to score the Masripithecus specimens and put them into this morphological, and it's actually a combined morphological and molecular matrix that was already set to go. And so we're running what are called Bayesian tip dating analyses. So until recently, the method really caught on, I guess in 2012 was when some of the seminal papers were published on Bayesian tip dating. So we ran our analyses with Masripithecus, and so Masripithecus was placed as an advanced stem hominoid. That means that it is closer to the living apes. It's still a stem ape. It's not within the crown group based on the Bayesian tip dating analyses, but it's more advanced along that stem lineage than any of the fossil species that are known from East Africa. The other important thing about Bayesian tip dating that I didn't note is that it provides estimates of divergence times, not just among the living species, but among the fossil species as well, based on this underlying clock, this evolutionary clock that is key to the whole method. So this provided an estimated divergence time of about 21 million years between Masripithecus and all of the other, all the crown apes within the Miocene. The Miocene started 23 million years ago. What we found for all of the other apes that have been found in Eastern Africa during the early Miocene is that those all split off of the stem lineage earlier. So actually not in the Miocene, but in the Oligocene. So multiple splits off of that stem lineage going back, 25, 27, 29 million years ago. The crown hominoids, many of the earliest crown hominoids are not from Africa. In fact, none of them are. They're from Europe, and they're from Asia, and they're from Turkey, Anatolia, which was a sort of a slightly different biogeographic region at the time. And so it's been a bit of a mystery why increased, or continuous sampling in East Africa has not produced an early Miocene species that is very clearly a member of that crown group. So that implies that these early Miocene lineages are somewhat archaic lineages that have persisted in East Africa into the early Miocene, and in some cases maybe even into the middle Miocene. And there's in fact a group called well, that had previously been called the Nyanzapithecines or Nyanzapithecids. Our analysis put a very mysterious ape that is known from the late Miocene of Italy called Australopithecus. It's always been a huge mystery because it combines features of crown features that are seen in crown anthropoids with other features that are really primitive and teeth that look like these Nyanzapithecids from East Africa. Puts Australopithecus deep into that clade, and that clade as a whole would have branched off, maybe 28 million or 26 million. I can't remember the exact estimate in Africa, and then Australopithecus must have made it into Europe much more recently in the late Miocene. So that kind of helps to explain that weird part of the tree, and people I'm sure will. Some will disagree with that placement, but we'll see how it holds up with time. So once we had the output of the phylogenetic tree, we could then infer biogeographic patterns on the tree. And so the simplest way of doing that is to estimate ancestral states at each of the branching points throughout the tree, which is what we call nodes, like nodes on a tree, as plant biologists would refer to a tree. So you can simply assign species a score according to which continent they occur on, and then you can reconstruct the ancestral states. So whether assign Afro-Arabia to all these early African forms, Europe, Asia, and then we added in a special category for Anatolia, Turkey, because it's a slightly uncertain geographic situation for that particular landmass. And so we found that all of the earliest split points in hominoid evolution along the stem lineage occurred in Africa, very unequivocally, going all the way back to the earliest stem catarrhines, earliest stem hominoids, cercopithecus. That all definitely is African, and it's unequivocally African. But then when we get to that node where Masripithecus is branching off from the other more recent crown hominoids, it gets to be much more equivocal. So you start to get mixed reconstructions that suggest that there is some possibility that these nodes, these common ancestors, might have been in some kind of a transitional zone between Africa, Europe, and Asia. And so what we ended up doing, because those simple reconstructions were not very satisfying, so we came up with a new method that we think is more useful. Basically, we took a 6x6 grid, which we put over a map of Africa, Europe, and Asia, basically encompassing all of the areas where fossil species in the matrix have been found. And then we reconstructed ancestral estimates at each of these nodes for those latitudinal and longitudinal blocks. And so that allowed us to get what are called posterior probabilities for each of the latitudinal gradients and each of the longitudinal gradients. We could get posterior probabilities, and then since we could get these posterior probabilities for both longitude and latitude, we could then calculate a joint probability for each of those cells. So what that allowed us to do, and this is maybe a little bit too much, but what that allowed us to do is estimate the odds of these ancestors being in any part of that grid. And so what we found is that for crown hominoids, that the highest odds on that grid were in northern Africa, into the Levant, and northern Arabia. And when you look at other parts of that grid, the probability or the odds is basically zero. So, like in East Africa, the odds of finding an early crown hominoid, according to this analysis, is zero. When we go farther back, we estimated a number of different nodes. So we looked at the initial split between apes and monkeys, and that was unequivocally East African. So the odds are very high in East Africa and basically zero everywhere else. We also estimated the odds for the origin of the so-called great apes and humans. So the clade that includes orangutans, chimps, gorillas, and humans. And that node turned out to also be the odds were highest in northern Africa, a little bit more northern into the Levant and into the Caucasus and kind of southeastern Europe. So it really, the analysis really strongly suggests that that group, that the living apes, are more likely centered in that sort of transitional area. And what I don't think I've even noted yet, is that Africa and Arabia were not connected to Europe or Asia until a ways into the early Miocene. So Africa and Arabia had been separate from Europe and Asia for going back like 100 million years ago. And they started to make a connection with Europe and Asia into the early Miocene. So very slowly they started to make contact, largely in the region that is now Iraq and kind of into Iran. This is a lot of the collision zone. And so starting about 18 million years ago, the what was called the Tethys Sea that was separating Africa and Arabia from Europe and Asia started to shrink, and finally there were land connections. And so right around that time is when we're getting that estimate for the age of what we call the crown hominid node. This is the great apes and humans right around 18 million years ago. And so the implication is that these groups right at the base of the living apes were more likely in that northern Arabia Levant region. And when when Africa and Arabia made their final connection to Europe and Asia, they were able to immediately spread out and head out to take advantage of the different environments that were available in those on those different continents. And indeed, what seems to happen is that very shortly after that connection is made, it's very poor evidence, but there are isolated teeth that are known from Europe that post-date that by only 1 million years or 2 million years. So it does seem like it's all fairly consistent. One thing that I would like to point out is that Masripithecus is consistent with that scenario, but it is not key to that scenario being true. So if we take the same tree and remove Masripithecus, we get the same result, that the origin of the crown apes is going to be in northern Africa and northern Arabia. So, and all of this, of course, is relevant to human evolution, because what we see from our reconstruction of biogeography is that the African apes and humans might have gone through a phase when they were actually not in Africa. This is something that has been suggested by several people before, notably David Begun and others, that there is a migration back into Africa, um, potentially in the earliest part of the late Miocene, and that migration back into Africa led to the diversification of gorillas, chimps, and humans, and all of those splits into different geographic regions. So all of these steps that we're reconstructing are ultimately that they are anatomical changes and geographic changes that are ultimately leading up to the origin of the lineage that would give rise to humans. So all of this work is very relevant to that question.
The senior author of the paper is Professor Hesham Sallam of the Sallam Lab. You've co authored quite a few publications on primate fossils from Egypt with Professor Sallam. What people may not know is that he was a student of yours back in your Oxford days here in England. It's quite an incredible journey that you both had, isn't it?
Yeah, it really is. So Hisham came to Oxford in 2007. He was one of my first graduate students. I accepted two PhD students that year. And yeah, so he had come in on a full scholarship from the Egyptian government. And the expectation of his scholarship is that he would get his PhD and then he would come back to Egypt and that he would start a vertebrate paleontology center, and he would basically lead the development of vertebrate paleontology in Egypt for the rest of his career. And so it's been a long and fascinating journey. The many aspects of Hisham's journey that I think he could write a really fascinating book about, and much of that is his story to tell. But yeah, I mean, shortly after he arrived at Oxford, I decided to leave to go to Stony Brook, which was quite a shock for him to a year after he arrived at Oxford, his advisor is disappearing. But ultimately, it was for the best because he ended up coming to Stony Brook. I mean, he got his DPhil at Oxford, but he came to Stony Brook for multiple years to work with me in my lab. And Stony Brook was a real, as I mentioned, is a real vertebrate paleontology powerhouse with a lot of really important people working on different topics. So it was really good for Hisham that he got to meet a lot of people in different parts of vertebrate paleontology. We were able to go on field trips into the Cretaceous of Egypt and Kenya, where he learned a lot about other types of vertebrates like dinosaurs and crocodiles. And that would be really important for some of the initial work that he would do when he came back to Egypt, when he made a spectacular discovery of a dinosaur in Egypt that made a huge splash and turned out to be really exciting. But yes, we're continuing our work at BQ2. When Hisham returned to Egypt after many years away, he discovered that BQ2 had been largely destroyed. It had been turned into a tomato field by a local farmer. So a lot of fossils were lost, no doubt, but miraculously, the most fossiliferous part of BQ2, which is a relatively small part, was left intact. And so what Hisham has been doing with his students and his volunteers over the last many years, because we don't really know, how long we have to continue that work there, is that he has been going in and jacketing. So it's called plaster jacketing, actually taking out chunks of the quarry and bringing them back to his lab at Mansoura so that they can be worked through very carefully under a microscope in a much more controlled setting. Oftentimes at BQ2, it's extremely windy and somewhat chaotic, and they're tiny little fossils in the matrix there. So it's difficult to do the work. So now he's gone there with kids of all ages. He sends me pictures from the field of kids who are maybe 7, 8, 10 years old, who are helping him to create these jackets that he brings back to Mansoura. There's still an enormous amount to be found in those jackets. There's going to be no doubt hundreds of primate fossils in those jackets yet to be excavated. And so that's fantastic, but it's also just really wonderful to see how much vertebrate paleontology has grown in Egypt in the last few years under his leadership, not only as being a mentor for all of these students who are now getting their PhDs in different universities, but also he, over and over and over, he is taking these young Egyptians who never would have had exposure to that to vertebrate paleontology on that level, involving them in the field work. And he sends these pictures of all these kids, just huge smiles on their faces, deeply involved in this work, and it's just providing a totally new perspective for them on the world they live on, the evolution that has happened through time, and just I think in a greater appreciation for the natural world that I think is going to be, we'll feel the repercussions of that in the next many years and decades, as hopefully those kids go on to be interested in evolutionary biology or paleontology, or wherever they end up. They have that perspective that I think is so important for the future of our species and for the future of this planet that we really desperately need to take better care of.
Well, it really is fascinating work that you and your colleagues are doing, Eric, and I'm very grateful you've been able to take the time to come on to the show today. So, what's new for you? What upcoming projects can you tell us about?
So right now I'm involved primarily in three projects in three different countries. So there's the work in Egypt that has been ongoing for a very long time. I'm also involved in a project in the Turkana Basin of Kenya at a site called Topernawi, which is a really spectacular site that is about 29.5 million years in age, and seems to be documenting the initial diversification of apes and monkeys in Africa, where I think we're getting at that split between hominoids and the old world monkeys. And also have a project in the Amazon basin of Peru in an area that's called Santa Rosa. And this is a site that is yielding primate fossils, probably the earliest stem platyrine monkeys, some of which are remarkably primitive and are going to have, I think, a really important impact on our understanding of convergent evolution of features between the catarrhine anthropoids and the platyrrhine anthropoids. So those are the three major field projects that I'm involved in. Unfortunately, I have probably 150 papers or more that I still need to write based on all of the discoveries that we've made over the years. I probably will not be able to finish those all up before I pass away, but I'll do my best. And I'm also finishing up a book on evolution and consciousness, which is a very, very, very different avenue that I've taken recently, but I think will hopefully also be of great general interest to people.
Professor ERIK Seiffert, thank you very much indeed.
Thank you, Mark. I really enjoyed it, and again, I really appreciate that you're doing this. It's incredibly important work.